Good morning everyone and welcome back to the class of Application of Biotechnology. Students, in the previous video, we have started the second chapter of Unit 4, that is Application of Biotechnology, and in that I taught you the application of biotechnology in the field of agriculture, where we saw about the Bacillus thuringiensis and we saw about the BT cotton, BT corn, and I told you how we can prevent our crops from these pests. Okay, so that first portion was completed. Now we have one more pest resistant technology, or we can say that pest resistant crop, and that is tobacco plant. Now we can obtain pest resistant plant by using pest resistant plant by using a method named as RNA interference. RNA interference technique. Okay, commonly called as RNAi technique. Now see, what is this technique? Children, all the eukaryotes, they have a self-defense mechanism in the form of dsRNA. And while using this principle, this concept, the scientists have developed the RNAi technique. That is, all the eukaryotes, they have self-defense mechanism in the form of dsRNA. Now we will see what is this technique. We are talking about the pest-resistant plant, and this we study, this RNAi, we studied in case of tobacco plant. And there is one insect named as Hirodine incognita. It is a nematoda, okay, named as Hirodine incognita. This nematoda generally infects the tobacco plant. Okay, it eats the leaf and it infects the tobacco plant. Now we want the tobacco plant which should be free from the nematode. It should be free. So we need one pest resistant plant. The plant in which there is no effect of the nematoda, that is Hirodine incognita. Okay, generally in the traditional method what we do, we put some chemical insecticides to kill these insects. But actually these things are harmful for the crops. So we need a resistant plant which can fight by itself against the infection or against the destruction caused by the nematoda. So just to develop the technique, what the scientists have done, scientists have made a disease-free, or we can say that pest-resistant plant. Now how they did this? See the mechanism. Now children, in case of nematoda, okay, I am going to erase this and I will draw . So our technology is RNA interference technique. This technique was used by the scientists to get pest-free or pest-resistant tobacco plant. Okay, and this was caused by nematoda Now no need of writing, otherwise I'll use all the space. Again I have to erase something. So see what the scientists have done. Basically in nematoda, they produce the gene, and this gene undergo for transcription and translation. Okay, we all know that this will undergo for transcription, and after transcription this gene will form mRNA, normal mRNA. Okay, now what happens next? This mRNA will again undergo for the process of translation, and then what it will form? It will form the required protein, and this protein is important for the growth of nematoda. This is a normal and general process, children. Okay, that is nematoda. They all have the gene. This gene undergo for the transcription and translation, and the gene produces protein, and this protein is required for the growth of nematoda. It's a normal step. Now what the scientists have done? The scientists have taken the gene from the nematoda body. They have taken the gene, okay, and introduced this gene. They have taken the gene, this gene, and introduced it into tobacco plant. This is important. This was done by the scientist. Simply they have introduced this gene into the tobacco plant. Now in tobacco plant, this gene again undergone for transcription. Okay, undergone for transcription, and then it produces double-stranded RNA, that is dsRNA. This was the step. Now come to, this is the whole chart that the scientists have done. Now children, as this nematoda will produce mRNA in the body, okay, for the general growth, what this nematoda will do? This nematoda will feed on the tobacco plant. Okay, so for the regular growth of the body, nematoda will feed on tobacco plant. And as soon as the nematode will feed on the tobacco plant, this mRNA will be silenced by dsRNA. Okay, dsRNA will identify that this mRNA is of its own body, because the dsRNA is from the nematoda's body only. So this dsRNA will identify the mRNA and it will bind, and this dsRNA will silence the mRNA, and because of this silencing, mRNA will not be able to undergo for the process of the translation. There will be no protein formation. Okay, and if no protein formation, then at last the nematode will die. Okay, nematoda will die. This is the silenced mechanism by the dsRNA, and that's why it is called as the RNA interference. Understood? So this is the concept. Now see once again what has happened here. Scientists, they took the gene from the nematode body, introduced this gene into the tobacco plant. Inside the tobacco plant, the gene has developed and it forms dsRNA. while the normal transcription process, it forms the double-stranded RNA. At the same time, inside the body of the nematoda also, the gene will undergo for the transcription. It will form the mRNA. And when this nematode will feed on the tobacco plant, then this mRNA will be bound by the dsRNA of the same gene. Okay? And this dsRNA will silence the functioning of the mRNA, and hence mRNA will not be able to produce, it will not be able to undergo for the process of translation and for the formation of the protein. And hence the nematode will not get the substance for the growth of its body, and ultimately the nematoda will die. So this is the concept of RNA interference technique to control the pest in case of tobacco plant. Okay, so this much is okay. Now our next point is Hirodine. Okay, I am going to erase this. It's better you all can take the screenshot of this, so that with the help of these notes only you can make the notes and you can prepare the things. Now children, we have one more concept, and your one more concept is Hirodine. Okay, Hirodine, and this, for Hirodine we use the plant named as Brassica napus. Okay, Brassica napus. Now see what happens in this case. Hirodine is an anticoagulant. Clear? And this Hirodine is found in the saliva of leech. Important point to note down. What is this Hirodine? This Hirodine is an anticoagulant which stops the blood clotting. Anticoagulant, you know the meaning of anticoagulant. And this is found in the saliva of the leech. Now children, why we want this anticoagulant? Okay, why we need this? Most of the time we go for the blood test and all. Okay, and the doctor and these people, lab technicians, they used to take the blood, and our blood should not go dried up. It should not dry, just to prevent them for drying, so that they can do the test. Just to prevent them from drying, we use one anticoagulant. Okay, they used to put one anticoagulant so that our blood remains in the liquid form and they can go for the further test. Now, from where they have got this anticoagulant? This anticoagulant is found in the saliva of the leech. Now, what the scientists have done? Now, I think so the points are almost clear that what we do in case of biotechnology, how we obtain the products. So, imagine what the scientists must have done for this. They must have taken the gene from the saliva of the leech. So, what the scientists have done? They took the gene of Hirodine, because it is an anticoagulant. From where? From the saliva of leech, and introduced this into one plant named as Brassica napus. Okay, named as Brassica napus. Now, what happens? This plant grows up. Okay, and along with this, the gene will also grow. Gene will also develop. It will undergo for the process of, naturally you all know very well now, for the process of transcription, translation, and it will form the protein. It will form the Hirodine, and that will be stored in the seed of the napus plant. in the seed of Brassica napus. And what the scientist will do? The scientist will take the oil from the seed, and this can be used as anticoagulant. Understood? That oil can be used as anticoagulant. So this is the second important application in today's class. Okay, one we have already done. So altogether 3 applications we have seen. So this is the second application in today's class, that is about the Hirodine, and this Hirodine is an anticoagulant found in the saliva of the leech. The scientists have taken the gene and introduced the gene into Brassica napus. In the Brassica napus, it will undergo for the normal transcription and translation, and the protein will be formed, and this protein will be stored in the seed of the Brassica napus. Now what the scientists will do? They will take the seed and they will take the oil of the seed, and that seed oil will be used as an anticoagulant. Understood? So this is all about the today's application of biotechnology in case of plants. Next, I think so we have 1 or 2 applications more. One is flavor saver tomatoes and one is to enhance the nutritional value. This we will see in the next class. Thank you, students.